As Janine just mentioned, I'm going to talk to you today about how at Indipro we're leveraging proximity to develop novel bispecific antibodies. And so, as our name implies, we are very interested in proximity, not only induced proximity, but inherent proximity. And we think about this in two different ways, and we try to leverage both of these. So, inherent proximity is really referring to proteins that reside in similar protein neighborhoods or microenvironments. And although membranes are very fluid and proteins move in and out and are very dynamic, there still are these protein neighborhoods, particularly in things like lipid rafts, and we believe strongly that these areas constitute a lot of shared biology as well. We also think about induced proximity, and there it's really modulating the composition of reagents to drive novel biology and signaling in those cases. And today I'm really going to focus on the aspects of our pipeline that are associated with inherent proximity. And so how do we look at inherent proximity at Indipro? I have to thank Jim Wells here for giving a very, very nice introduction in the photocatalytic labeling platform that was pioneered by two of the co-founders of Indipro while they were at Merck. And we've used that. And we found that it's really a huge advancement over more conventional approaches towards cell surface labeling. So as you may be familiar, if you use an enzyme to label the surface of proteins on the surface of cells, and that enzyme might be tethered to an antibody that binds to a target of interest, because the half-life of those enzymes tends to be quite long, you end up labeling a very, very broad surface that covers the cell. If you use a photocatalyst, however, you get really, really discrete resolution, and this is really driven by the short half-life that's associated with these different photocatalysts. So you can see in the cartoon there that this a photocatalyst can be, again, covalently bound to an antibody that binds to a target of interest, and depending on the photocatalyst that's used, when you expose that to visible light, you generate a very highly reactive intermediate that can then covalently bind to proteins within that proximal neighborhood. And can you hear me okay? Okay. And so if you look at the example below there, we're showing that in a co-culture system where we were using T cells and antigen-presenting cells, we used an antibody that bound to PD-L1 that didn't block PD-1, PD-L1 engagement, that was tethered with the photocatalyst. And you can see 3 different volcano plots on the bottom where it's the proteins in the upper right-hand quadrant that are enriched following this labeling technique. And if we use 2 different photocatalysts, either a short or mid-range label, you can see a series of proteins that are emerging. And with the short-range photocatalyst, we really just see PD-1 or PD-L1, which is the target that we labeled. With a mid-range catalyst, now you start to see a broader range of proteins. And again, you can label proteins in cis as well as in trans using this approach. And importantly, if you compare that to the volcano plot on the far right, which is an example of using an enzyme for labeling, you get this very large series of proteins that are very, very difficult to really resolve what's happening with respect to proximity. So this system is very tunable. It doesn't perturb cells. We can use this type of a system on live cells, on frozen tissue, in a variety of different ways. And I'm just highlighting here in this slide basically what we've described as our protein neighborhood mapping toolbox. So we have a number of different photocatalytic probes. They have different characteristics such that some may bind agnostically to any amino acid residue. Some can bind specifically to things like a tyrosine residue, and you can see there that they have different half-lives, and they end up labeling different spatial environments, all within the narrow range of a few hundred nanometers. And some of these photocatalysts are very well suited for labeling within cell culture systems and organoid models. On the other hand, there are another set of photocatalysts that can be have slightly longer half-lives, have a broader labeling radius, and those are more amenable toward labeling more complex tissues, such as frozen tumor tissues, et cetera. And so we can apply all of these different approaches to really start to define proximity mapping and neighborhoods on different cell surfaces. And there are many different applications to this photocatalytic labeling. As I've just described, there's this tunable distance labeling that we can do. if there's concern that an antibody is too bulky and may disrupt the inherent architecture of proximity of different proteins, you can label directly. You can also use this by tethering a photocatalyst to a ligand to deorphanize receptors. You can also use this in a primary and secondary labeling component, as you can see on the upper right-hand side there. And as I mentioned already, we can label both proteins in cis and in trans in a co-culture system. And we're also able to identify intracellular proteins that maybe constitute different signaling components that are also in proximity or might be brought into proximity when you have two different co-culture systems and ligands and receptors engage one another. And we also do a lot of cell surface proteomics, and so you can apply all of these using the photocatalytic labels. And so we don't just do one, when we do this, we call this technique micromapping. And as I showed you, the data emerges as a volcano plot, so we've deconvoluted this using mass spectrometry, and this is how we pull this together. But we don't just run one micromap experiment on a target of interest. In fact, we run dozens. We run them on dozens of different types of cell systems or tissues. And doing this, we have been able to start to develop and characterize these very, very large surface proteome networks over the time that Indipro has been in business, which has been a couple of years. And we can also start to apply this to different types of classes and indications of proteins, and we have particular interest in receptor tyrosine kinases and immune receptors and tumor antigens as well. And so collectively, using AI ML, we're building very, very large data sets that are allowing us to really start to characterize proximity networks on different types of cell surfaces and under different conditions of cell activation states as well. And so since this technology was pioneered, and while we've been using it at Indipro, we've really industrialized this entire process. So this highlights here, illustrates the workflow scheme that we start with, where we may have an indication of interest, or we may have particular tumor antigens that we are interested in targeting. We identify the cell systems that we want to use. This could be a whole range of cancer cells, primary tissues, disaggregated tissues from tumors, et cetera. And then we also spend a lot of time validating the probes that we're going to use, which are generally antibodies, but we do a lot of antibody validation, and then we run a large number of these micromap experiments, and we put them through in our database, and I'll describe this to you further in the next few slides, where we generate these knowledge graphs. And we use this as an approach to look for and identify novel target co-pairs that may be amenable for future therapeutics, and these can really apply to a number of different modalities. And so we've spent a lot of time really industrializing this platform over the last couple of years. We use far fewer numbers of primary cells, and we can use many high throughput approaches also to run these experiments rapidly and to characterize and analyze the data more rapidly as well. And so here again is sort of how we consolidate all of this data. We call this database MINT, for membrane interactomics. And going into this database is a lot of our proprietary data from the micromapping experiments that I've just described, the cell surface proteomics data that we generate as well. But we also layer into here a lot of data from publicly available data sets, which may correspond to tumor versus normal expression, information about different signaling networks, and so on. And all of this basically gets consolidated and is displayed on what we call a knowledge graph, which is really just like a t-SNE plot. It's taking all of this multidimensional data that you see on the left-hand side there and presenting it in a two-dimensional space. And out of these knowledge graphs, then, we evaluate these novel co-target pairings in an unbiased manner, and we apply this towards selecting, ideally, co-target pairs to generate reagents and tools against and understand whether they have therapeutic value. And so it's really not just proximity alone that's important to make a good bispecific therapeutic. There are many, many other characteristics which we try to build into this database, which is continually learning. The more micromaps we run, the more data we have to go from. And so as we've already heard several times already in this conference, particularly in oncology, one of the challenges is that there are many modalities but very few targets. And over the last 25 years, the field has really continued to work on pretty much all of the same tumor antigens, although they may be applied with different modalities or with novel linker payloads or engineered into T-cell engagers and so on. There's really a dearth of tumor antigen targets that we've worked on. And we really believe that by leveraging this photocatalytic labeling and micromapping approach, not only are we identifying novel target co-pairs for known tumor antigens, but we can also then take a novel target that we've identified, micromap it, and develop bispecifics that are comprised of two novel targets. And as I mentioned again, proximity alone is not going to be the driver for developing a good therapeutic, but all of the other aspects that we've applied into our database help us to try to down select what target co-pairs to look at. And so one of the tumor antigens that we have looked at extensively is EGFR. And so we really asked this question, can we make a better EGFR molecule by generating a novel target co-pair? And so as we all know, we've been studying EGFR for a very, very long time. And most, although there are many approved EGFR or TKI therapies, they're really only partially effective. And this is largely due to the fact that EGFR, as we all know, is quite broadly expressed, and this restricts the utility of use across many different indications. And it prevents really expanding to having a better therapeutic index. And so we think if we make a bispecific ADC that's comprised of EGFR and another target, which we call a TAPA, which stands for a tumor-associated proximity antigen, we can generate a bispecific molecule that will boost that selectivity, therefore expand the patient populations this can be used in, and we're really leveraging this shared biology. So I should have mentioned this earlier, but one of the things we really believe about proteins that reside in these neighborhoods and have inherent proximity, they very frequently have a shared biology, and that's really something that we think is important to tap into and ultimately can translate into a more potent therapeutic. So we started with EGFR, and we performed the micromapping experiments that I mentioned here across a number of different cell systems, including cancer cell lines, frozen tumors, primary cells as well. And out of this, we identified a protein called CDCP1, where once again, I need to thank Jim Wells for the very elegant introduction, not only to photocatalytic labeling yesterday, but also to the CDCP1 protein. And so we've done reciprocal micromapping experiments as well, where we can target CDCP1, and we can also easily pull out EGFR as being inherently proximal. And not only do we just pull out CDCP1, we see, as you can see in that expansion of the knowledge graph there where EGFR is highlighted, there are a number of targets that emerge. Some are known and common, and others are less well known. But throughout all of that, we selected CDCP1 as the highest target of interest in order to proceed forward with. So just a little bit more about CDCP1. CDCP1, that stands for Cub Domain Containing Protein. It was discovered as a tumor-associated antigen with a poorly defined function. As you can see in the graph on the bottom right there, it's highly expressed across a broad range of tumor types. And it's also been demonstrated to actually form a ternary complex with SRC and EGFR that can activate downstream signaling. And downstream of that, the CDCP1 molecule seems to be quite important in driving tumor cell migration, particularly of cancer cells into a metastatic niche. It's also highly expressed and regulated by activated RAS. It's upregulated when RAS is present. It's associated with poor overall survival in a number of different cancers and poor clinical outcomes. And it's also been implicated in being a driver of resistance to TKI therapies as well as chemotherapies. And so here's just an example of CDCP1 expression in lung adenocarcinoma and squamous lung carcinoma, indicating its association with poor progression. So if you look at these Kaplan-Meier curves on the left for overall survival, on the right for recurrence-free survival, you can see that those patients that have high levels of CDCP1, which is illustrated in the red lines in all of these curves, you can see they have reduced overall survival compared to those patients that have lower CDCP1 expression. And so we did extensive amounts of immunohistochemical staining to look at EGFR and CDCP1 co-expression, and I'm only showing a very small summary of that data here. And the graph on the left-hand side is color-coded such that those regions in each bar that have the darkest blue show the highest degree of target co-expression. And then as the colors become lighter, those are a range of high-high, high-low, low-high, et cetera, et cetera. But you can see just by looking at the colors that there's high co-expression across a broad range of different tumor types. And the examples on the right-hand side just show some examples such as lung, bladder, head and neck cancer as well, where we see very, very high co-expression. And if you look at those corresponding normal tissues, we really don't see much expression. With the exception of skin, where we know there's a lot of EGFR already expressed, we see some CDCP1 there, and I'll address that in the next few slides. So also, if we just look more broadly at CDCP1 expression across normal tissues, and we compare that to the expression of other very known and well-used tumor antigens, you can see on the lower part in the front there that CDCP1, its expression in normal tissues, it's quite restricted. And so with all of this knowledge, then we embarked on a campaign to look for novel CDCP1 binders, and we looked for VHH binders. And so I just want to walk you through some of the approaches that we take when we're looking for novel binders. And so in this case, we had a number of CDCP1s that were just paired with FC and were screened in internalization assays. And these were stratified based on different epitope bins. And so the internalization assay data on the left-hand side of the graph just shows that there were a series of binders that corresponded to one particular epitope that really drove internalization much better than the other epitope from bin B shown on the bottom. And we had already committed to wanting to explore this as an antibody-drug conjugate, so we were really favoring selecting a binder that drove internalization better than others. If you wanted to make a T-cell engager out of this bispecific EGFR-CDCP1 pair, we would have selected, and in fact, we've done this as well, one of those in the bin B molecules. And so we took these CDCP1 binder VHHs that showed high internalization, and we paired those with a number of different EGFR binders that corresponded to a range of EGFR binding affinity. And then we applied those into additional internalization assays where we used wild-type SK-HEP1 cells, which express high levels of both EGFR and CDCP1, or cells where we had knocked out CDCP1. And that data is shown on the right-hand side there. And so you can see that the different colored symbols correspond to different EGFR binders that have a different affinity. And if you just focus on those pink symbols there, you can see that there's really not a lot of difference in terms of selectivity of internalization, whether or not CDCP1 is present. But in contrast with those green symbols, we still see high internalization in the wild-type cells, but much reduced in the CDCP1 knockout cells. And it's really that cluster of bispecifics that we were interested in, and we're really trying to tune the affinity of both of these binders to favor internalization and therefore killing of target cells when both targets are present. And so we did a lot of detuning and exploration of EGFR binding affinity, obviously because it's the more abundantly expressed target across normal tissues, and we maintained high CDCP1 affinity. And just want to show you here that we were actually able to detune EGFR to as low as 500 nanomolar. And in the context of the bispecific, we were able to see very, very strong avidity binding effects. And so here's just an example of a different binder where if you only put EGFR or only CDCP1 or both targets down on a chip, you can see the binding curves shown there. And in the context of having both targets on the chip, you can see a very pronounced a binding activity when both targets are present, even when the EGFR binder is very, very low affinity. And so here's a little bit of the description of our bispecific molecule. So we've really tried to optimize the molecule in many different ways to make a first-in-class bispecific ADC. So as I've already described to you, we maintained the binding affinity of EGFR. This is a Fab, very low. This binder also does not impinge on EGF signaling, and we think that in normal tissues where EGF signaling is important, we want to maintain that signaling process. We think this will improve the overall tolerability of the molecule. The CDCP1 binder that we selected is a very potent internalizer. It also has high affinity. We have silenced the FC not only to minimize hinge proteolysis, but to also potentially minimize engagement with FC receptors in tissues such as the lung and the liver, which has Kupffer cells that may contribute to reduced tolerability of the molecule. And we've engineered this as an MMAE with a very low DAR. It's a DAR 2 on engineered cysteines, and we've made this as a topo inhibitor as well, but I won't be speaking about that molecule today. So we've characterized IDP001 quite extensively, and through the selection process, we've always used a variety of different cell lines that correspond to a range of EGFR and CDCP1 expression, as you can see in the examples on the right-hand side there. And so all of these graphs here are just showing that IDP001 shows substantially enhanced internalization, either against the EGFR or CDCP1 mAb that was derived from those binders, or to competitor EGFR or CDCP1 bivalent antibodies that are in clinical development or already approved. And so in each of these graphs, these are internalization assays. We're looking at internalization over time. IDP001 is in the bright blue line, and then the monospecific molecules are in the other lines. And you can also see, if you just look at PC3 as the example, it's one of the few cell lines where EGFR expression tends to be lower than CDCP1, and there again we see very nice internalization with IDP001 and very weak internalization with the EGFR binders and also with the CDCP1 bivalent binder as well. And so, as I mentioned earlier, there is some expression of CDCP1 in skin, and we've explored that as well. And if you just look at the histograms on the left-hand side there, you can see that there is similar co-expression on, in this case, H1975 cells, which are a lung adenocarcinoma cell, compared to primary lung fibroblasts. However, when we perform micromapping experiments on normal tissue versus tumor tissue, we see that there is reduced enrichment or reduced proximity in the normal tissues compared to the tumor tissues. And so that's shown in the graph with red and blue bars there, where we have, again, this differential localization across those tissues. And if we look in vitro viability assays using either primary cells, keratinocytes, or lung fibroblasts, we see dramatically reduced killing compared to the killing that we see on tumor cells. And similarly, we see reduced internalization on the primary cells compared to tumor cells. So we think what this is telling us is that the spatial proximity or the inherent proximity of EGFR and CDCP1 is probably quite different on primary cells, or at least on these keratinocytes that we've looked at, compared to the tumor cells. And this will also help to drive a broader therapeutic window with this bispecific over more conventional EGFR antibody therapies. And as Jim alluded to yesterday too, this protein CDCP1 can be cleaved, but upon cleavage, it really stays associated. The cleaved domain stays associated. And in fact, that's a cartoon from one of Jim's publications earlier that described that. And so we asked whether or not IDP001 was still functional when CDCP1 was cleaved, and in order to do that, we used a few cell lines where we either treated them with plasmin, which will cleave CDCP1. You can see that in the Western blot there, in the two cell lines HCC827 is another line where CDCP1 is endogenously cleaved. And in all of those, we found that we still had functional activity as demonstrated by the viability curves on the right-hand side there. And IDP001's activity was really not influenced by the cleavage of CDCP1. So we've looked at CDCP1 activity in vivo, and these are just a number of different xenograft models, where again, as I mentioned previously, we really look at a range of EGFR and CDCP1 expression, because we recognize there's going to be broad heterogeneity in any patient population that we go into. And we've used these types of studies, similar to the cell-based internalization assays, to understand where's the sweet spot where we can get maximum internalization and killing of tumor cells, but still spare primary cells. And so with all of these different xenograft models here, you can see we have very nice anti-tumor activity with just a single IV dose administration. And if we use just a CDCP1 or an EGFR monovalent binder in the graphs on the right-hand side there, we still have activity, but it's not as good as the bispecific. And if we look in patient-derived xenograft models, and these are all lung patient-derived xenograft models, which also correspond to a range of EGFR or RAS mutations in them, again, using a single dose IV administration of IDP001, largely at 3 mg per kg, in one model also at 1 mg per kg, we see very nice tumor growth inhibition activity. So we're really excited about IDP001. It's a first-in-class bispecific ADC. It has very low affinity EGFR binding compared to any other EGFR therapies that are in the clinic, and it maintains signaling downstream of EGF as well. We've optimized it for ADC activity as well as for tolerability. We have potent in vivo efficacy data, and we've completed all our non-human primate studies where IDP001 has been shown to be very, very well tolerated. So we're advancing this into the clinic early next year, and we're very excited about that. And so we've applied this photocatalytic labeling and micromapping approach not only to EGFR, but to many, many other well-known tumor antigens. And we're really using this technology, which we've really now industrialized, to define different tumor proximity landscapes using our MINT database. And we're really excited about how this can open up the field and open up identification of additional novel targets that can either be paired with known tumor antigens or co-paired in novel settings to really expand the toolbox for generating bispecific ADCs to treat cancer. And so I'll just close by acknowledging the team. It's always a team effort, and the folks at the MINT platform there, which was originally pioneered by Rob and me and brought into IndiPro, has been terrific. We have an excellent biologics team where we're able to make all of our bispecifics in-house in a high throughput manner, screen them in a high throughput manner, and this allows us to really explore a variety of different formats and ranges of affinities to down select for the ideal molecules. And so I'll stop here and thank you for your time.
Hi, Pam. Really great talk. I think your platform is very elegant, and I appreciate you taking us through it today. I did have a question. You mentioned the CDCP1 you had an epitope that gave you the internalization that you really were looking for. I'm just curious if that's at all related to the cleavage phenomenon that you guys also see in that same protein, and if at all that might be related.
Yeah, I don't know that it is related because, again, even when CDCP1 is cleaved, it creates this novel neoepitope, which is very interesting. But the whole molecule stays largely intact. And we think that what's driving that enhanced internalization, again, is the inherent proximity of those two molecules together.
Okay, cool. Thank you.
Hey,
Jonathan?
My battery's being changed. So.
Yeah. Okay, then we go over there. And very quick, because we are running out of time.
Sure, great talk. I just wonder if you have mentioned, like, if CDCP1 actually binds to EGFR, do they interact? And the second is, you mentioned in the normal tissues, you see less association between these two molecules. I'm wondering what's your interpretation for that? Do you think it's just because in the cancer cells, in the RAS-driven model, you have upregulated CDCP1, so that's why in the normal tissues you don't see this association or neighborhood that much?
So to your first question, others have shown that they can co-immunoprecipitate EGFR and CDCP1. We really haven't done those experiments ourselves because the proximity distances that we see would suggest that even if they're not physically co-associated, they're very high in proximity. And as I tried to show, there's much less CDCP1 expression in the normal tissues, and the way we've engineered this molecule is to selectively internalize. So it's not exclusive 100% that it can't get in if CDCP1 is absent or EGFR, but you can see there's a dramatic difference, and that was even in the xenograft data where we use a monospecific binders, where there's still some tumor growth inhibition, but it's nowhere near as potent as the bispecific. Does that answer your question?
Okay.
Very quick, Jonathan.
Very quick, yes. So thank you. Really interesting talk. A lot of ideas to talk about. But what I'm just curious about is if you've worked out the average amount of labeling you'd get based on concentrations of the two different components that you're looking at, and how much that plays a role. So if you have if you label one guy and then there's a lot of something else, there's more chance of it being close, even if there's not any actual reason for it to be close. So do you have a way to sort of control for that and look for a change that's beyond what you would expect from randomness?
Are you referring to the actual labeling and the micromapping itself?
Yeah, to do the mapping, yes.
So again, it's just, you mean how much antibody we add to bind?
Well, you add some antibody, but then you get a signal how many, what different proteins got labeled by the, reactive species. But if there's a protein that there's 5 million copies of it, some of it will be labeled just because there's so much of it, right? So there's some sort of whereas if there's 10,000 copies and a bunch of that's labeled, it means a lot, right?
Yeah, so I think what you're asking is there a difference that's related to basically the protein abundance at the start of the experiment? Mm-hmm. Not really, and I'm not the mass spectrometrist, I'm not the chemist who does those types of experiments, but we've looked at that specifically, and we are able to pull out binders from targets that have very low levels of expression in addition to those that have high levels of expression.
Right, but that would mean that the low ones are definitely close for a reason, right? They're not random.
Yes.
Okay.
Yeah, but we are able to pull those out as well.
Cool.
And there is some quantitative component to some of the mass spec work that follows, as well as the surface proteomics.
Okay, thank you.